It's 2 minutes past the hour, so let's just get started. Good morning, everyone. My name is Zhiyong Lu, a senior investigator at the National Library of Medicine Division of Intramural Research. On behalf of our division leadership, Dr. Richard Shorman and Dr. Dave Landsman, I would like to welcome you all today to our monthly LLM Colloquia on biomedical data science and computational biology research lecture series. So this series brings leading scientists and researchers from across the biomedical and computational science to share their insights and wisdom with us that advance discovery and innovation in many ways that touches our life and in health and medicine. So we are so honored to have with us today Dr. Tiarnán Keenan, who is a Stadman tenure-track investigator at the National Eye Institute, NEI. As a clinician scientist on campus, Dr. Keenan sees patients as a retinal specialist, while leading research focused on retinal diseases, in particular age-related macular degeneration, which is the leading cause of legal blindness in the US and beyond. Dr. Keenan received his undergrad and medical degrees from University of Oxford in the UK, and then completed his academic clinical training in ophthalmology and biomedical research at Manchester Royal Eye Hospital and Oxford Eye Hospital. He later came to the US as a Fulbright scholar. So I have had, over the years, I have had the fortunate to work very closely with Dr. Keenan through our long-time collaboration with NEI, and I've been truly impressed with his exceptional research ability to bridge clinical insights with advanced computational and AI approaches. A few years ago, he was invited to brief the US Congress on AI applications in retinal diseases, which I think he's going to talk to us more about it in the next 45 minutes. So without further ado, please join me in welcoming Dr. Keenan.
Thanks. Yeah. Thank you so much for that really generous introduction, and thank you to the leadership here for the invitation to speak at this colloquia. It's a great privilege to be part of this fantastic series. So just see. Okay, so I can advance the slides. Yeah. So as you heard, on the clinical side, I see patients with retinal disease, and one of the diseases I see most frequently is macular degeneration, which, as you heard, is the leading cause of blindness in the US. And that's my main research interest also. And I've several kind of interconnected areas of research in macular and retinal disease. On the one hand, more traditional clinical research, clinical trials. We have a lot of interest in, oral supplement, nutritional dietary approaches, which are really important in macular degeneration. And I'm not going to talk so much about that today. But then on the other areas, ophthalmology and retinal disease is very kind of highly image-centric. And so we have really rich data sets, of imaging. And so, alongside that, I'm lucky to work with, imaging experts, computer scientists, statisticians. And so it's that area I'm going to focus more on today, kind of statistical, deep learning, and computational approaches to trying to, improve our diagnosis and our treatment of retinal disease. But I should say I don't have a specifically computational background. I have a clinical background, and then my PhD was actually in, basic science, biochemistry, and genetics. But I'm really lucky to work with, with fantastic people in this kind of interdisciplinary area, as I say. So I should say I have no conflicts of interest to disclose. So what I thought I'd do today is approach the talk in this kind of way. Because quite a few of the applications I'll talk about are related to macular degeneration, I thought I should start with just kind of 4 or 5 minutes, a bit of an introduction to what we mean by AMD, and then use that to say a quick word about why I think ophthalmology is particularly well suited to deep learning approaches and AI more generally. And then so if the if the title here was diverse applications, what I did was, from our large body of work, kind of choose a few hopefully representative projects that I've kind of, listed there, 1 to 4, all touching on macular degeneration in different ways. And they're roughly chronological, moving from kind of older ones to, at the end, much more recent unpublished work. So some dealing more with kind of computer vision type tasks, some related to, how do we approach a new potential biomarker in retinal disease, both statistically and computationally, and then the third and fourth, I'm trying to think specifically about clinical trials. So I hope there'll be something of interest to most people. And I was thinking originally about talking about cataract, but I don't think I'm going to have time, so I'm just going to focus on the back of the eye. Okay, so yeah, maybe just starting with a bit of an introduction to macular degeneration. Well, essentially, it's really important because it's common, it's costly, and it's increasing. So certainly in the United States, and actually in all industrialized countries, it's by far the leading cause of blindness and visual impairment. But then even globally also, it's massively increasing as populations age. And the thing with AMD is that it's, as the name implies, its risk increases with age. In fact, it increases truly exponentially. So by the time we get to about 60 years and older, it's as common as, in fact, more than 1 in 4 people. So it's incredibly common. In the US, that corresponds to about 20 million people living with AMD. And globally, that number is 200 million, but it's predicted to get to closer to 300 million in the next 15 years or so. So it's kind of a hidden problem, but it is actually a massive public health issue. And that's, of course, because this is the kind of functional impact that macular degeneration has in its advanced stages. So the macula, is the central retina responsible for our central high acuity vision. And in advanced stages, then, patients, undergo kind of profound central blindness. So kind of maintaining navigational vision, but losing the ability to read, to recognize faces, to drive. And so there's a massive kind of knock-on health impact in terms of loss of independence, falls, depression, healthcare-associated costs. And this is what it looks like when we take photographs of the back of the eye at the various different stages of macular degeneration. We don't see the clinical manifestations until after the age of about 50 or so. And then very slowly over the course of, maybe a couple of decades, it can advance through these stages, becoming more symptomatic until you get to the kind of advanced disease at the right there, being either wet or advanced dry disease, so-called geographic atrophy. And then, I think I've got some higher magnification images of each of these stages that I'll just run through fairly quickly. So early AMD is actually really pretty subtle. If we look at the macula, you can almost miss it, but there are these small deposits called drusen, these kind of yellowish extracellular, mostly lipid-related deposits. And so there are millions of people with early AMD, who don't know about it. It's undiagnosed because it's asymptomatic. And this is why screening, is really important, because there are things we can do to slow progression. And let's say if we kind of fast forward 10, 15 years, now people would be at the intermediate AMD stage, and still the symptoms are fairly mild, and visual acuity is typically fairly good at this stage, close to 20/20 perhaps, but the drusen now, these deposits are much bigger, more numerous, and so we diagnose and stage AMD through the kind of drusen size and characteristics. And you get some other imaging features as well at this stage. But the important part... Oh, yeah. Sorry, why biochemically is... Yeah, so the extracellular deposits, they're mostly lipid-containing, but they have a lot of other molecules from the innate immune system. So for example, on the genetic side, we know that one of the main drivers of AMD is the complement part of the innate immune system. And so indeed, they have a lot of kind of complement-related molecules and complement regulators. So, and I can certainly delve into that if that's helpful. But yeah, both from biochemistry and genetics, we know that it's quite an immunologically driven disease with this very long preclinical phase, occurring for many decades before we get to the point of these clinical manifestations. And then, at the point advanced disease occurs, like I said, that can go either, one of two ways, the so-called wet form, And that's shown here, particularly in the old days where you had no treatments available. It's kind of rapidly devastating to central vision. So abnormal blood vessels grow through underneath the retina. They leak profusely, cause massive scarring and hemorrhage, and very destructive to that very delicate tissue of the retina. And the other way that patients can go to advanced disease is a kind of dry degeneration called geographic atrophy, where you get a kind of geographic-looking patch of retina that's died. We see that kind of central depigmented patch in the color image. And so that corresponds to a profound blind spot. And the problem there is it starts off very small, but if you see this patients over the course of years, it kind of expands relentlessly. So this is some infrared reflectance imaging on one of our patients in the NEI clinic. And we see over the course of 2 or 3 years, that smaller patch just gets larger and larger over time. So equating to the death of all the photoreceptors in that area, the death of the accompanying retinal pigment epithelial cells, and there's a kind of profound area of central blindness that's expanding over time. And I'll come back to that later, especially in relation to some of the clinical trials. So that's a kind of overview of how we can think about AMD progression over the course of a few decades. One of the main challenges of AMD is that most of our existing treatments are available only at the advanced stages, and it's particularly true for wet AMD. And of course, that's when we've already started to have some irreversible vision loss. So, anti-VEGF injections, I'm showing an image there, for wet AMD were a massive step forward when they came along in the past 15 years or so, but they do have a lot of downsides. They need to be repeated really regularly, pretty much lifelong, often in both eyes. They're very costly, so it's a big burden for patients, for physicians, very costly, and all the monitoring that's required to go along with it. And then dry AMD, which is the majority of cases, we have actually very few treatments available so far. So, AMD, I think I mentioned the figure of 20 million people in the US. We've got a lot of other age-related diseases, glaucoma, cataract. So ophthalmology more generally has been, growing and growing in the demand with our kind of aging populations. So some data here from the UK showing it kind of outstripped a lot of the other common clinical specialties in terms of number of attendances. And in parallel with that, we've been lucky to have amazing advances in our imaging. So we have a lot of modalities these days, not just color photographs. We have 3-dimensional OCT imaging in pretty much every ophthalmologist office. We have increasingly high-resolution OCT that's almost cellular level resolution. Ultra-wide field imaging, really good for conditions like diabetes. OCT angiography, where you can image blood vessels without needing to give intravenous dye anymore. But altogether, this has meant we've kind of been experiencing an explosion in imaging. So it's increasingly the question is not can we image the retina in detail? We certainly can, but it's more, firstly, can we do it at scale? And secondly, can we do the more kind of refined quantitative evaluations that we'd like to do? And of course, where I'm leading to is that here is somewhere where I think AI can help. And there are other reasons also why I think AI is particularly well suited to ophthalmology. Like I said, we're really very much an imaging-centered specialty, alongside radiology, pathology. So there's a lot to be gained from computer vision approaches there. But also there's this increasing idea that the eye genuinely can act as a kind of window to the body. And this has spawned a whole new field called oculomics. And particularly in relation to the cardiovascular system and the central nervous system, there's great excitement with the idea that we may be able to use retinal imaging to predict risk of cardiovascular disease and risk of dementia, Alzheimer's, and other neurodegenerative diseases. Because, of course, in the retina, we have a unique situation where we've got part of the CNS and a microvascular circulation directly visualizable with our complex imaging modalities these days. So I'm not going to talk about that so much, but we do have a major collaboration plan between the NEI and the Center for Alzheimer's-Related Research, the CARD clinic, in that kind of oculomics field. And in fact, ophthalmology has been right at the forefront of clinical AI from the beginning. The first FDA-approved autonomous AI system in any branch of medicine was actually in ophthalmology, and that was this system I'm showing at the bottom here that performs automated detection of diabetic retinopathy. But importantly, it does that through being deployed in primary care offices where you have patients with diabetes coming through. You don't have ophthalmologists there. So this approach in a few seconds can pick up some diabetic retinopathy, and patients can get to see an ophthalmologist that way. And I think hopefully AMD is going to be the next frontier because of that scale and that kind of that complexity. So in our group and with others and with Dr. Liu, we spend a lot of time thinking about how AI can support AMD care and kind of throughout the clinical care pathway, because the kind of AI approaches you might need in screening would be different to those that would be useful in a retina specialist clinic or from those that would help us in clinical trials. Okay, so yeah, that's by way of a kind of long introduction to set the scene a little bit, and then I thought I would delve into a few examples of some of the work we've done in this area at different parts of that AMD care workflow. Yeah, so the first, and this is a little older now, using convolutional neural networks, but it was a kind of one of the first major projects we did in collaboration with Dr. Liu and his team to try and use deep learning to help us with diagnostic and prognostic tasks in AMD. So as Dr. Liu was mentioning, we've been really fortunate to have this collaboration for many years now, I think back since even like 2017, 18, and on the NEI side, led particularly by Dr. Emily Chu, and then we've been lucky to work with some fantastic postdocs in Dr. Liu's lab. I've kind of alluded a bit to this already, but why would one be interested in doing this apart from just seeing if one can? Firstly, accuracy is one part of it. So it's always fun if you can see if the deep learning model beats the retina specialist. That's great, but I think probably the more important part is that second part. Can we reach all these millions of undiagnosed people and get them to kind of have greatly reduced progression to advanced AMD to prevent that irreversible vision loss? So that second part, I think, is the more exciting. So as I say, this was a few years ago now, but but we wanted to see if we could get neural networks to try and kind of replicate the human clinical workflow that we go through when we see a patient in clinic that we may suspect has macular degeneration. And so the patient in front of us in the clinic, we imagine here, is kind of represented by a pair of color fundus photographs. And because we've run, very large multi-center trials for kind of, 5 or 10 years at a time, we have many tens of thousands of images like this with a whole spectrum of AMD severity and features. And then not only that, because, that's how these trials were run, every single one of these thousands of images has had really detailed grading for all kinds of AMD features at these centralized image grading centers that we have in ophthalmology. So I won't dwell on the details too much, but in the kind of conventional way, we had a kind of training and a test split. So we trained the algorithms to see if they could detect AMD and all its constituent features using these labels, and then we had a holdout test set. And then rather than just reporting the performance of the algorithm, in a kind of vacuum, we were able to compare it to the performance of experts because we had the performance of these 88 retina specialists on the same test set. I think one question that often comes up is, should you have kind of more end-to-end training, a bit more of a kind of black box approach, or should you have a kind of more transparent approach? And we went for that latter approach in this workflow because we used each of the neural networks as individual biomarker detectors. And that way, you have a bit more transparency and potentially for clinicians and patients, a bit more kind of, trust and explainability in the system. So we had one trained to detect drusen and to do drusen size grading, one for another important feature, pigmentary abnormalities, one for late AMD, which of course we have to pick up. And then, as you've probably seen in many specialties in, dermatology, cardiology, et cetera, the neural networks can not only meet but often surpass the performance of the retina specialists. And so they do that for each feature typically, and they can therefore do it overall. And so that's kind of considering things at a certain moment in time, but probably what's more interesting and exciting is can we use neural networks to kind of help us look into the future in the form of personalization. So, we have someone with some degree of AMD in the clinic. We really want to know, what's going to happen in 5 years or 10 years, and how can that inform the treatment decisions I want to make. And so that's what we try to do here in this extension work. And again, we have the patient in front of us in the clinic represented by a pair of color fundus photographs. And We can give a little bit of additional information. We maybe give age, maybe smoking status. They're important for AMD progression. But we're going to ask back from the algorithm, a personalized prediction. So what is the percentage of this, percentage risk of this individual developing advanced disease over the coming years? We may ask kind of over 1 year, and we may ask over kind of 5 or 10 years. And again, to kind of compare this to our existing clinical standards, we have a couple of clinical standards that can do these kind of predictions to a degree. We have one really quite simple approach called the arid simple scale. It makes these quite coarse predictions over a single fixed 5-year period. We have a slightly more complex approach, an online calculator that can give you more continuous predictions. But of course, both of these approaches are totally reliant on the clinician seeing the patient and entering the drusen and feature characteristics. So actually, we know that our accuracy in those assessments is not great. And then for the deep learning workflow, this is how we approach the problem. We kind of broke it into two. So on the left, we've got the patient in front of us represented by a pair of fundus photographs. Then we have the neural networks extracting the features of interest. And then those are fed in the second part of the approach into a survival model, more of a traditional statistical approach to return to us the percentage risk. And at that second stage, we can provide or we can withhold additional information like complex genetics, et cetera. But we had two different iterations, and again, this gets back to that question of transparency, versus black box. We had one so-called deep learning grading, where we used only features that we already knew were kind of traditional risk features. But then the second one, so-called deep feature extraction, we allowed the neural networks to extract whatever features they thought were most predictive of progression, and there could be many more of them. There could be 20 plus. And on the plus side, you might get some increased accuracy, but then, of course, on the downside, you might lose some transparency. We may not necessarily be able to give those features all a name. And so that was the workflow. We had a large heterogeneous data set, and all these thousands of individuals, we knew who had indeed progressed to advanced AMD and when they had done it. Was it at 1 year or 10 years? So that made for a good data set for training and a challenging data set for the test set. And there are a lot of details I could go into. I've just tried to summarize the main results in one table here. Essentially, on the holdout test set, when we ask either our clinical standards or the neural network for 5-year predictions, and we've got the two human clinical standards on the top there, and we've got the two totally automated deep learning approaches on the bottom. These are the kind of performance statistics we see. And specifically, we're measuring the accuracy of the predictions using the C statistic, which is a bit like the AUROC for predictions. So 100 means your predictions lined up perfectly with the order in which people did indeed progress to late AMD, and 50, of course, would be not better than random. And so again, as you might imagine, we see, the human experts using these calculators get some reasonable predictions in the region of 81, 82, and then our totally automated approaches not only meet, but actually they significantly surpass the accuracy of those approaches. And as I say, that's without any genetics. All they get is the images, age, and smoking status. So again, they're doing this very accurately. And similar idea in external validation and the same pattern of results on every year of predictions we ask them to make.
Tiarnán, sorry, I'm going to miss this, but what exactly are you predicting? Regression, yes or no, or is there some quantitative value
Yeah.
predicting?
So we're asking for a kind of, do I have it here somewhere? The percentage risk. So if we have an individual in front of us who does not yet have advanced AMD, in the clinic, we want to know, is that person likely to progress in 1 year or 10 years? So we ask from both the kind of clinical systems and the deep learning systems, give us a percentage of that individual developing advanced AMD and we ask at every interval, 1 year, 5 years, 10 years.
Yeah. So advanced AMD, yes or no?
Yeah, and a percentage risk specifically. Yeah, and then we compare the order of the percentages with the order in which people did or did not progress to late AMD over time. Yeah, exactly. I wasn't going to talk about the genetics too much. If it's interesting at the end, I can come back and say what does it mean to provide detailed genetic information as well. But in the interest of time, I was going to move on, staying a little bit with that theme about trying to predict the risk of progression to late AMD, which is what we're spending a lot of time doing in the clinic, but thinking about those were traditional biomarkers, drusen pigmentary abnormalities. What does it mean if we have a new potential biomarker? And can we use deep learning approaches to help us, and statistical approaches also? So if those were the traditional features, there's this much more recently appreciated imaging feature called pseudodrusen, reticular pseudodrusen. And pseudodrusen, of course, just because they're not kind of like traditional drusen, they're actually in a different layer in the retina. And the important thing about them is that they've only become appreciated much more recently, because we basically don't really see them when we perform normal clinical examination or take normal color pictures. When we start to perform more complex, more recent imaging modalities, then we start to appreciate this feature. So people have been puzzled, what can it mean? There's a dramatic example here I'm showing on the right. If we do fundus autofluorescence, so we excite the retina at a certain wavelength and then capture the fluorescence signal by taking the image at a different wavelength, we see sometimes these striking kind of zebra-like pattern feature, so-called reticular pseudodrusen. And then we also see it on the OCT, the three-dimensional image, and we can distinguish them from true drusen. But essentially the question had come up, does this represent an additional risk feature? Because it seemed like people with these RPD were quite likely to progress to late AMD, but it wasn't clear, is it just because they also happen to have a lot of normal drusen as well? And as I say, the main difficulty was that with our normal color imaging, we don't tend to see them. We need to turn to these more advanced imaging modalities. So there's a detailed method slide here, but I just have one point to make. So we have these really large kind of historical records, the AREDS 1 and the AREDS 2, these massive multi-center studies of many thousands of people with AMD followed for many years. AREDS 2 did include, because it was a bit more recent, some of the newer imaging modalities like fundus autofluorescence, so we can grade RPD. But to understand RPD, this new feature, we really needed to go back to AREDS 1 because that had the full spectrum of baseline AMD severity, and follow-up was for as long as 10 years. But that's, being an older study, it had only the simple color fundus imaging, the traditional imaging, where we can't see RPD. So we turned to deep learning essentially to see if it might be possible to kind of infer the presence of this new feature on color fundus photographs, even if human grading is not able to do that. And again, this was one of our early kind of landmark projects with Dr. Liu's group, and again, a lot of the fantastic postdocs, particularly Chinyu Chen, were working on this with me back then. But essentially, the way we were able to try and approach this was because through AREDS 2, the more recent study, we did have all imaging modalities available, both the fundus autofluorescence and the color images. So we had, I think, over 8,000 instances of these multimodal image pairs. And I think at least, in retina and probably in radiology and other specialties as well, this multimodal imaging with label transfer can be really powerful because, in this case, for example, we had all of the, many thousands of FAF images graded for RPD. But then, of course, that label can be applied not just to those images, but to the corresponding color image pairs. And then we can feed, thousands of color fundus images into the neural network, and the accompanying labels for RPD are going to be accurate even though they came from the other modality. And so that's the way we approached it here. We tried a few different neural network architectures. And it turned out you can train deep learning models to kind of infer this feature, even if it's not visible to expert human graders. And I know attention maps have their limitations, but it seemed they were doing it really plausibly. They were seeming to pay attention to parts of the image that did indeed have RPD if you look at the corresponding fundus autofluorescence image. And we improved the accuracy further by using multimodal multitask training. And I think I won't dwell on that too much, but essentially the main difference there was in the previous approach, when we were trying to train an algorithm that worked only on color fundus photographs, and that's all that was present during training. But in this approach, even if we were trying to train an algorithm that worked only on color fundus photographs, both modalities were present during the training. And it seemed like that kind of increased improved the shared representation, the fact that both modalities were present during training. So then we had improved performance. I think on external validation, it had close to kind of 0.98 AUROC on a challenging test set. So essentially, we could take the algorithm that was trained on a more recent data set that had both modalities available, and we can now apply it to a much older, kind of crucially important historical data set that had only the traditional imaging available, and a study that can never really be repeated, at that scale or that size, AREDS1. And so, I think the specific, clinical conclusions are maybe less relevant here, but as it happened, it turned out RPD is a really important risk factor. Its presence doubles the risk of progression to late AMD. And so if you have a lot of traditional drusen and pigmented abnormalities, your risk is already quite high. If additionally you have RPD, you can take that pre-existing risk and double it. So we showed that it was a risk feature. It's, even in combination with these other features that we did know about. And it increased the risk of late AMD at all baseline severity levels, and it increased the risk of both subtypes of late AMD. As I say, the specific clinical conclusions may be less important, but I hope this is a nice example of how deep learning algorithms can add a whole new dimension to a kind of crucial historical data set by inferring the presence of a particular feature that's only more recently become appreciated on some new imaging modality. So as it happened, that was important, so that we could develop a new classification system for AMD. And then more recently, that was looking at RPD at a kind of binary level in the image. More recent work, we're trying to do more like individual pixel level segmentation of RPD. What does it mean to have a massive amount of RPD versus only a small amount? That's probably important, but we need these kind of approaches to curate data sets at scale to be able to answer those kind of questions. I think I maybe won't say so much about the second part. This is more on the biostatistical side, but RPD also became really interesting to people looking at geographic atrophy. So once you have geographic atrophy, I showed those images of it expanding across the macula. That expansion is much faster in some people and much slower in others, and that's important also for clinical trials. So people have thought maybe RPD is a risk factor for much faster expansion, and it became complex because some people thought, well, maybe it is, but probably only as a proxy for genotype, specifically this one really important risk locus on chromosome 10. So in this work, we showed that it was an important risk factor for faster GA growth, and then we used mediation analysis to kind of decompose this complex triangular relationship to show it genuinely as a causal, it's an independent and a causal risk factor, and separately, ARMS2HTRA1, this important risk locus, is also associated with faster GA growth, and RPD is not a risk factor purely as a proxy. So I can return to that, but I think maybe for this audience, I'll move on to a couple of more recent applications. And so this is staying with AMD, but focusing a bit more on that geographic atrophy. Like I showed those images of the atrophy expanding across the macula. And as I said, that rate of expansion is really important in clinic, for giving people prognosis. We're trying to give people an idea of whether or not they should be having treatment to try and slow the expansion rate. And then definitely in the area of clinical trials, there's a massive amount of work and time and money spent on clinical trials these days trying to develop treatments that slow the geographic atrophy progression rate. And so we're always trying to see if we could design our clinical trials more efficiently. And this work was done principally by a great biostatistician in my group called Jintong Hu, who's just joined in the past year or so. And yeah, as I say, this is really recent work we're just about to submit. So I mentioned some of this already, but many, many trials, and we've had a lot of failures, a couple of recent FDA approvals for trials with kind of modest success, but we're trying to slow down that expansion rate of geographic atrophy. For a long time, it seemed like it was just inevitable and relentless, but we've had a couple of modest successes recently. So there's a lot of interest in trying to develop treatments to slow down that growth rate. The trouble is, this is a really slow rate of progression. The rates vary a lot between different people. So that means we need really massive trials, like many, many hundreds of people followed for a couple of years to try and show, a partial slowing in its expansion rate. So in this work, we thought, can we approach this, a bit more intelligently? Particularly, can we use this property of geographic atrophy whereby, although the growth rates vary a lot between different eyes, for any given eye, the growth rate over time tends to remain pretty similar. So it expands in quite a linear, monotonic way. And so if we could use this in some clever way, it meant we could have still highly powered trials, but with many fewer participants. or shorter trials, or both. And because they're just, a limited pool of participants, so it's really prohibitive at the moment, both in terms of people and costs. So I'm sure you're really familiar with the traditional, randomized controlled trials. So, in geographic atrophy, you enroll, many hundreds of people with geographic atrophy. You randomize them at baseline. Let's say half of them are going to get the real treatment, half will get the placebo or sham. You follow them and repeat the imaging over the course of 2 years. And at the end, you hope there's, a difference with the ones allocated to the real treatment showing a slower growth rate. And so we just thought, what would it mean to make one small difference here? We're still going to do a randomized controlled trial, but we're going to try and add in a run-in phase. So you still enroll all the individuals with geographic atrophy, but you don't randomize straight away. You just wait a short period, let's say 3 months or so. And so you image at baseline, you image again at 3 months, and that way you may be just passing out a bit of the heterogeneity in the GA growth rates between individuals. And just for 3 months, and then you proceed with your RCT as normal, you randomize, you follow people for the rest of the trial. And so the idea is if we take those GA growth rates that collected during that 3-month natural history period, and we feed those into the outcome vector for the statistical model, maybe it might help increase the power or decrease the sample size requirement for the same power. And then we'll come to that when we model this and see if that is the case or not. And then the second approach is a little bit similar, but even more extreme, in fact. So this trial design, we actually don't randomize people at all. So we enroll everyone like before with geographic atrophy. We again just follow them to collect natural history information, maybe for 4 or 6 months this time. And then at that point in the middle, as I say, there's no randomization, but everyone begins treatment. So you enter the treatment phase, everyone gets treatment for the year or so, and then you image again at the end. And this is really attractive to patients because, in the traditional RCT, you may go through all of this period, and it turns out, you only had a half chance to get any treatment at all, effective or not. Whereas this type of trial, everyone gets treatment. But so the idea that's clever here is that basically each eye is serving as its own control. So, if the GA growth rates are pretty steep during the first phase, and then generally much flatter during the treatment phase, if we pass that out statistically, that's going to give us a strong treatment effect readout. And then on the methods side, on the one part, we used more traditional statistical approaches to perform, power sample size analyses. So, if we want those parameters in the middle, let's say we want a trial with 90% power to show a 25% treatment effect or greater, and the trial's going to last 2 years, we'll get a readout of what sample size we need. But then I think that's not enough increasingly. So what we did in the second part of this workflow is, we get that, let's say we need 200 people according to that calculation. We then take, a sample size of 200, and then we run simulations, specifically Monte Carlo simulations. So it's like we've performed kind of 10,000 of these trials. So we perform many replications, a little bit different every time, but that way we can kind of empirically validate what is the power, is it genuinely 90%, and what's the kind of false positive rate, the alpha, and to kind of make sure that the results from those traditional power analyses are indeed validated empirically. And then I think we've got a massive amount of work and many hundreds of thousands of simulated clinical trials to feed into. I tried to make it just one simple table like this. And in fact, we can make it simpler by just concentrating on one column. Maybe if we take the right-hand column. So what we've got here are the 3 different designs shown on each row. So at the top, we've got a traditional RCT. In the middle, we've got RCTs, but adding in run-in phases of different duration. And then at the bottom, we've got the more extreme trial, the single-arm one. And in each case, we've got the required sample size to give us 90% power to get that treatment effect. So you can see with the traditional approach, we needed just over 150 patients. So, and that means 75 are going to go to active treatment, the other half randomized to placebo. But you can see what happens when we add in just a short run-in phase. And importantly, we don't increase the overall duration of the trial. If we add in a 3-month run-in phase, we subtract that from the post-randomization phase. And so we can decrease the sample size by as much as almost 50%, down to kind of 82 or so, just by having that 3 months where we waited and collected some natural history data, and we fed that into the outcome vector for the statistical model. And I think it is because we're just parsing a bit of that heterogeneity between growth rates. That's why it's so helpful. And the longer you're willing to wait in the run-in phase, the more powerful that effect is. Maybe 9 months is a bit long because GA will be progressing during that period, but 3 months, 4 months, maybe even 6 months, I think is very reasonable. And now we can do still really powerful trials with much a smaller, or you could go for a shorter trial as well. You can see it's even more extreme when we are willing to go to a single-arm trial. And I think this has probably worked for phase 2, probably not for phase 3. I think you need randomization in phase 3. But there, with as few as 14 people, it turns out you can have 90% power to show a treatment effect, just because of that really powerful idea where each eye is acting as its own control. So this wouldn't be possible without kind of computational approaches, both for our images with geographic atrophy, and then being able to run these thousands of simulations for every small change that we make. We've done other things I won't dwell on too much. What does it mean to have like 2-to-1 or 3-to-1 randomization instead of 1-to-1? It turns out that you can increase power that way as well. But in particular, I just wanted to show an example how we can use computational approaches on the imaging side, and then some machine learning and statistical approaches on the clinical trial simulation side to try and make some progress, design the next generation of clinical trials in AMD. And then if we have a few minutes before the end, I'll maybe just say a quick word along slightly similar lines. Staying with the idea of geographic atrophy, but thinking about new structural outcome measures, because so much of the success or failure of our clinical trials is dependent on the trial design and what outcome measure we pick. Like generally in ophthalmology, so much what we do is related to visual acuity. That's the kind of crucial, quality of life measure. But it turns out that in geographic atrophy, it's very difficult to use visual acuity, and it's very difficult to predict visual acuity directly from retinal images. Someone with atrophy like that, if you measure acuity one day, it'll be very different if you measure it again 2 days later. So it turns out it's a very difficult metric to use in clinical trials. So what I was trying to do here was derive a new structural outcome measure that tracks visual acuity, which is our key functional measure. And this is recent work from my group, and we need a lot of different expertise on the image analysis side, on the computational side, on the biostatistical side. So each of these members kind of came together to make this project possible. So because visual acuity, is so noisy, has that massive test-retest variability, it's not used in trials. So all the GA trials going on these days use GA area. And that's nice because it's objective. You can measure its increase over time. It's a simple number, but it has this massive problem that it assumes all macular locations are equally important, whereas we know for visual acuity and for quality of life, that's absolutely not the case. That the central macula makes a massive contribution to visual acuity, the peripheral macula very little, but we don't take that into account. So we may have drugs approved, they're actually not that helpful, or equally we may have drugs fail because GA area didn't pick up what's actually important to patients. So I wanted to see if we could design an outcome measure that could bridge these two, ideally have the advantages of both. And we can scale it from 0 to 100, so that's what I did for simplicity. So I wanted it to be an outcome measure that could be read directly from images, like a structural outcome measure. It's objective, but it would change. It would kind of scale from 0 to 100 in parallel with changes in visual acuity. And essentially, the way that we approached that was to divide the macula into kind of concentric rings. And I think I've got a slide on the left here, showing, we chose 60 rings for our primary work, but we varied the numbers, so any number you like really could be okay. And we can imagine that, these rings near the center of the macula, we're going to need to weight them more highly for this new metric, and the ones near the peripheral macula, we're going to probably need to weight them in a lower way. They make much less contribution to visual acuity. But we want to do that not arbitrarily, we want to do that empirically. And again, through these trials that we've run, we've got many thousands of instances of images with geographic atrophy, all shapes and sizes, and we have the corresponding visual acuity measurement. So even though it's noisy for an individual, we should be able to derive a metric that, that overcomes that problem. And so when we, for each of these images, divided the macula into 60 concentric rings, we can calculate the GA occupancy of each annulus. And then to derive the weights of each of the rings, we used ridge regression. We tried a lot of different regression-based approaches, but that's a machine learning approach that's really useful when you have a lot of inputs, each of which are very correlated with each other, which is certainly the case for these GA occupancies. And then when we've derived the weights, we can calculate the final metric, which I call here Gawain, as the weighted sum of all of the rings. And then we can use a series of outcome measures to make sure it is indeed closely associated with geographic atrophy as it should be. And so these are the kind of images that we have available, all shapes and sizes of GA. We can overlay the rings, calculate the GA occupancy, and we can do that computationally. So these are the main results for the weightings. If you look at the figure on the right and concentrate on the blue dots, going from left to right on the x-axis, these are the rings from the center. As we might imagine, they were indeed really highly weighted. And then when you get further out, the weighting becomes almost negligible. The contribution of those peripheral macular areas to visual acuity is minimal at a certain point. And changing the annulus number doesn't make too much difference. The results of the weights almost fall along that same regression line. So I won't dwell on this, but it turned out, this new index was really well correlated with visual acuity, both cross-sectionally and then also longitudinally. Change in this new index was really well correlated with change in visual acuity over time. And according to whatever imaging modality you had, it worked on color images and on those fundus autofluorescence images equally well. And then hopefully this will make it a bit more real and concrete. These are a couple of clinical examples. So in the top example, this is someone who has just a really tiny area of geographic atrophy, but it's right at the very center. And it turned out that even though it's only a tiny area of geographic atrophy, because those central rings are highly weighted, the Gawain was quite high. It was 36 out of 100. And in parallel with that, the best corrected visual acuity was decreased by a kind of equivalent amount. And then the contrasting example on the bottom there, you've got a much larger area of GA shown with that kind of red hatching. But because they're all non-central, actually, the weighting is very low, so the Gawain is tiny. It's only 2 out of 100. And in parallel with that, the visual acuity is barely decreased at all. And then that tracks over time. If you follow those cases longitudinally, the visual acuity is shown in the charts in kind of reddish color. I think that's jumping around all over the place like it does, which makes it just impossible to use in clinical trials. But meanwhile, the Gawain is increasing in a kind of much smoother way that makes it potentially really useful to power trials. So I think that's, all the examples I wanted to give. I wanted to try and give some from, more like diagnostic prognostic tasks and some clinical trial-orientated ones. How do we do with new imaging features, and how do we use these approaches to try and help us with new structural outcome measures? So, yeah, again, I'm really grateful to the collaboration we've had with the NLM, particularly Dr. Liu's lab over the past many years now. So on our NEI side, as I say, that was Dr. Chu and colleagues. And then we've been lucky to work with many of the great postdocs shown here, including current postdocs. And then some of the members of my group here I'm showing on the left. So, yeah, and finally, thank you again for the invitation, and thank you for your attention.
Session of the presentation. So for those of you who are in the room, please press on your mic before you ask questions. And then for people online, if you're joining us online, you can send your questions to NLM Colloquia at nih.gov, NLM COLLOQUIA at nih.gov. Thanks. Richard.
Yeah, thanks, Tiarnán. That was really very interesting. For your third section with this kind of trial design project, where you have the period before treatment and you use that as kind of establishing your baseline rates, I guess. In reality, you're kind of comparing that to historical data because it's based on the assumption that the rate is monotonic and it's going to continue to increase. So how does the FDA view that kind of an approach? Because that is kind of what you're comparing against, this historical data, right?
Yeah, there've been one or two trials that have used run-in periods, but they, at least in my opinion, haven't used them to the maximum, utility. So one or two have used them for stratification during the randomization. So let's say 3 or 4 months, they look at GA growth rates, and then there's one that kind of binarized them to like faster versus slower GA growth rates and used that as a stratification factor during randomization. But, I think that adds maybe a little bit to the power, but I think, this kind of approach would add massive amount more to the power. Like, and we tried to, what does it mean to calculate the GA growth rate during that period and to use it as a covariate in the model? That was a bit helpful, but actually the most powerful thing was feeding those data directly into the outcome vector for the model. So the FDA are very supportive of covariate adjustment. They actually recommended it in a report a couple of years ago. So I think maybe in the old days, there was a very pure randomization. You just take all comers, you randomize, you let that deal with it. You don't do covariate adjustment. But I think there's been a move in the past few years in the community and specifically also in the FDA to, we can maximize the power of these trials and we can have smaller trials by doing covariate adjustment.
So I guess that's when you still have the 2 arms. But if you have the single arm, there you're really relying on this monotonic growth rate.
Yeah, that's true. And so again, there have been 1 or 2 trials. So actually, we ran one ourselves that was a single-arm trial that so I used some of the parameters of that for these simulations. As it happens, it was a failed trial. It was oral minocycline to try and inhibit microglial activation to see if that would slow GA growth rates. It didn't, but as a proof of principle, I think it worked really well. And I think for phase 2, it's really probably perfect because there, I guess on the industry side, or if it's investigator-initiated, we can't have massive trials. There's only a limited amount of money. And so there, if you've got 10, 20, 30 participants, then perfect. And then that can give the kind of go, no-go decision for the phase 3 where you do need to randomize. So I think each of them has their kind of applicability in different settings. Yeah.
And the statistics is based on the Monte Carlo simulated data as your comparator?
Yeah, so we had kind of 2 parts to the workflow. One is using more traditional power analyses, and we try to, repeated measures regression, basically, which is what's usually used for these GA growth rates and the FDA likes. So we get a kind of more traditional statistical approach there, but then, the second part is to empirically validate those by doing Monte Carlo simulations, exactly. And in all cases, actually, the answers were really similar. But the simulations give additional information like the alpha as well, the false positive rate. So, yeah.
Thanks.
Let me ask a question for the online audience, and then we'll go back to
Speaking to the mic.
So there's a question. So you can see the question. Please comment on use of AI for smart glasses and ocular implants for macular degeneration.
Wow, no, it's a great question. Gosh, I'm not sure I have much to say there. Yes, smart glasses, I guess that's more like looking out. I mean, did I have an image somewhere towards the beginning of a kind of implant? So a lot of what we try to do in our work is to slow down the progression to advanced disease so you don't get that kind of very profound blindness. But then it's a different situation once people have, very established geographic atrophy with blindness. And then it becomes more kind of one of 2 things. On the one part, we and others are interested in, stem cell implants, so RPE stem cell technology. So our scientific director, Kapil Bharti, has a program there, potentially doing amazing work. But then the other side of things is we've got a certain amount of retina there. Can we maximize the functional vision for that, for that retina? And then things like, implanted chips or smart glasses or magnification, all these kind of approaches become much more applicable. So there's kind of, I guess, 2 aspects to it, trying to, maximize the health of the retina and then trying to work with the retina we've got to maximize functional vision. Yeah.
I have a question about your predictions of development of this severe AMD in the beginning. you showed the table with the predictions of like retina experts versus your neural network. So my question is, how predetermined is the development of the severe stage of AMD is? So you had like retina specialists predicting 82%, you predicting around 90. But if you had like a cohort of 1,000 people with exactly the same condition, will they all 100% of them develop this particular severe AMDs if you know if their condition is the same, or there is like natural variability?
No, it's a great question. And yeah, I think the short answer is no, that they won't, and that can be for a variety of different reasons. So I mentioned towards the beginning, like diet and oral supplementation are actually very, very profoundly important in AMD. So for the same kind of baseline characteristics and imaging features, you could, and same, I don't know, smoking status, you could have, varying trajectories whereby some people, let's say with very poor diet progress much, much more quickly, others much more slowly. I guess probably if you waited long enough, if everyone lived to 150, all of those 1,000 would get there in the end. So it's more about, the kind of the health span. Can the macula last the lifespan of the individual? So that's the way in which we tend to think about macular degeneration. Does that answer the question? Yeah.
Can you just add that the ground truth is actually
Right.
by reading centers, a third party, not by physicians?
Yeah. So in all these cases, we're really lucky in ophthalmology to have these centralized image grading centers. So and so for all of these studies, that provides the ground truth. So it's a kind of independent, and there are image graders that do nothing but go through these images, grade features, and have the appropriate software and workflows to do that. And then the clinicians are playing a slightly different role. Yeah.
So your work's largely about diagnostics, accurate diagnostics, and what are the classes of treatments that are available at this stage for AMD, and is it one class or is it many classes?
I'd say several classes, and it's very stage dependent. So if you get advanced wet AMD, by far and away the main class of treatment is blocking vascular endothelial growth factor, VEGF. And it turns out that just by blocking that one molecule, it's very effective. Of course, it's not a cure, and the injections need to be repeated because they leave the eye over the course of a month or two. But that has been probably the most profound step forward in AMD care ever. And so advanced geographic atrophy, dry disease with geographic atrophy, the main approaches that have been tried so far, and the two that have had FDA approval, are approaches that block the complement system, and the complement system biochemistry-wise and genetics-wise is clearly really important in AMD. They've had only modest success so far, so I think there are other neuroprotective approaches in trials that will probably be helpful. At the earlier stages, it's all about slowing progression, and it's more about lifestyle modification, and AREDS2 supplements are probably the main thing that has a big impact, and they're mostly antioxidant, and zinc as well, which may modulate the complement system. Yeah, so maybe we need to move to a system where we need not just stage-specific treatments, but maybe even genotype-specific ones. There are two main sets of genetic loci, one related to complement, but the other not actually related to a poorly understood factors at chromosome 10, probably to do with extracellular matrix turnover. But in the future, maybe we can have more success by more genotype-specific treatments as well. Yeah.
Okay, thank you. Thank you for wonderful talk. My question is related to David's. So in terms of association causality, do we know if, there's a relationship with autoimmune disease, and there is a great progress now in the treating of autoimmune disease. Are the treatment for autoimmune diseases that a person may have slowing down the progression of AMD?
No, that's a great thought, and we have looked into that in the early days in Oxford. I did a lot of epidemiology work. So, what does it mean for people to have, rheumatoid arthritis and to be on, DMARDs, and does that decrease or increase the risk of AMD? it does not make a massive difference. I mean, well, to start, firstly, we have a lot of analyses for genetic heterogeneity or pleiotropy, and it seems that AMD genetics is not highly overlapping with genetics of those other autoimmune diseases. It seems to be quite specific. So something quite special about the macula and the situation where you have an extracellular matrix in conjunction with a fenestrated capillary layer and then the photoreceptors. So, and then in those epidemiology studies, so conventional treatments for autoimmune diseases have not had a big impact, unfortunately, on decreasing progression to AMD. So it seems maybe, firstly, it's quite specifically, the complement system, and secondly, quite specifically, the location. So, and there are reasons for that, perhaps. Yeah, I mean, there's some overlap with some renal diseases and macular degeneration, but quite specific to do with complement factor H. Yeah.
Thank you. Thank you for this wonderful talk. It's and I appreciate you going through this whole body of work, spending 10 years of your life. It's truly impressive. I had a question. I noticed in the first part of your talk, as you were discussing the models, and one of the contribution is that you were able to identify and get a very accurate prediction based on the image of the eye, but also certain characteristics. Like you had in your slides that you had sex, age of the person, and smoking status and their BMI. But then on the second part of your talk, it was only the image. And I was wondering, especially there when we dropped the size of the trial to 14 people, are we able to account for the differences that were so important in the first part of the talk?
Yeah, no, that's a great point. And I guess there are two parts. There's the risk of developing advanced AMD, and we know pretty well the risk factors there, smoking, age, etc. And then separately, once you've got geographic atrophy and we're interested in the expansion rate, there's less research on the risk factors there. We've done quite a lot of work in that area, and there are some that are overlapping between the two stages, like smoking. There are some that are not, like the genetics. So, but in all cases, I think we should make use of all the available information from risk factors. So, but the beauty of the single-arm trial, for example, is in a way you don't need to worry for whatever risk factors they have to make their GA growth rate what it is. That then serves as a control, and if you've had a treatment effect, you'll see a kind of flattening of that growth rate. So, but yeah, I haven't dwelt on it, but we do tend to use covariate adjustment for whatever risk factors we're aware of, and not just imaging features, also demographic, medical, dietary, though that gets complex. Yeah, no, great question. Thank you.
Well, thanks very much.
Thanks so much. Thank you.
Wonderful talk, and thank you all for coming today. And just a reminder for some of the trainees in the room, you'll have another hour to spend with our speaker today, and you can ask any question. No PIs will be allowed there. Thank you very much. Have a great day.